epidemic, but also even more recently the yellow fever. Because we have the vaccine, it seems like people are a little bit, okay, we can rest and no further research. But also for an epidemic like yellow fever, but diagnostics is a key issue. Why? Not only for the rapid detection to allow for early intervention, but also during the epidemic. Again, the Ebola is a nice example, but it's the same for other pathogens as well. And then also after the epidemic, the monitoring, the surveillance, diagnostics play a key role. And yeah, I think many of us have worked in the field and have experienced the difficulties even making the difference between bacterial or viral infection. It is definitely not yet everywhere possible. So I'm excited about this panel. Thank you for being here. And so I would like to start launching the discussion a bit with the first question for your reaction. Is really, what do you think is needed in terms of network and infrastructure for ensuring rapid identification of epidemic-prone pathogens? So we're identification epidemic-prone pathogens. And also then, of course, considering the context of this workshop, how do you see the role of public-private platforms for diagnostics? Shall we start with Marie-Paul? You're the woman on the panel, so we have to stick together.
So thank you very much, and I'd like to thank the presenters, because actually it was both very lively and very interesting, and also putting the base to what I'd like to say right now. So, you know, we see that quite often there is not enough communication between the public health officials and the diagnostic industry on what are the tests which are needed. And this was exemplified with what was said in terms of Ebola. You know, that you don't need only one test. You need to have combination of tests in certain conditions. So for Zika, for example, we have been advocating. We have worked on a target product profile for diagnostics, which helps differentiate between different flavi, and this is not yet available. And we think this is so important right now. It's important for Zika, but it's also important for dengue. It's important. I have had some discussion with the colleagues from Sanofi Pasteur, 2 days before, for them, you know, seeing their dengue vaccine being rolled out in an area where there's no way to differentiate to see whether if people come with fever, they have Zika or they have dengue is an issue. So I think what would be good is to have a better dialogue on the definition of what tests are needed for public health use. Many things are possible, but this dialogue, I think, should be more brought to bear. The other need or infrastructure need that we have identified and on which we will be working in the coming months is responding to the problem that was mentioned also about the difficulty of testing these new diagnostic tests. In Ebola, it is the ethics review approval in Guinea. But why is that? It's also because there were no samples anywhere else. So everybody was, we have worked with quite a number of companies who were doing at the end when there were no cases anymore in many places except Sierra Leone at that time. But there was a long line of diagnostic to be tested in Sierra Leone, and very few samples, very few capacities. So what we would like to do, and I hope that this will help, is identify in different regions reference lab that can test for diagnostic and also equip this reference lab in advance with biological material corresponding to the priority diseases, at least that we have identified, a few more, so that they could get this material and have a biobank ready to go and do testing in advance of an epidemic. This is also responding to another thing that we have seen in another problem that we've seen in Brazil. So Brazil has a law that they cannot export serum or samples. So therefore, everybody is trying to get their sample tested in Brazil. No, WHO has an emergency listing procedure for Zika diagnostic open. We have had 20 applications, but it's only one test which has been able, we have been able to get through listing, because all the others are still waiting to be tested. So I believe that trying to work in advance, establishing in different regions reference lab with standard material might help kickstarting the testing of tests for an emergency.
Thank you very much. That's two very clear points. Piotr?
Thank you. For us, our experience comes from working mainly in public-private partnerships with IMI. And speaking about the role of public-private partnerships or platforms, what we hear when we engage in such a collaboration is from the national public health institutes that they have some concerns always about the scientific independence when you start working with in collaboration with the private sector. And although in Europe there is diversity between the approaches here, so there are countries where national public health institutes are quite used to working very closely with the commercial sector, with the industry. But there are some countries, and quite many, where there is no such collaboration, and actually in some countries it's prohibited by legislation. So here there is a risk that there could be the whole sector may be missing, you know, from such a collaboration. And from our experiences, again, mostly through the IMI Advanced project, the way in which it's being tried to be addressed is to develop some clear governance models for such public-private platforms, to very clearly define the roles of different stakeholders and in a very transparent way, and develop some kind of code of conduct, and actually starting with defining what is scientific independence, really, what are we talking about. Because, and then once this is developed, it remains to be seen how it works actually, how it will work through the advanced project. But then the communication strategy should be developed also to the public, because in many areas we may be in danger of losing the public trust. So the communication strategy of why the public side engages in such partnerships, what are the benefits, what are the how the transparency, scientific independence is guaranteed is quite important. So that's the kind of public side of this. Thank you.
Thank you, Piotr. Yeah, the scientific independence is a key point to be considered. Frank?
Well, I think I almost always, just said that. I think that an important, I actually agree with, the laboratories, all these class 3 and class 4 facilities. They're certainly important, also place to do clinical trials. But I still think, to actually try to combine information generated, at the frontline diagnostics with people that actually need to take action further back, I think is really key. So the sharing of data between, all the stakeholders, those generating data, but also those that actually need to take action, including academia, that are often the place where you see the new problems and the first characterization. I also think that we are missing out a lot in terms of the analytic capacities if we have this sometimes a little bit structured collection of data and information. There are a lot of smart people out there, especially in the bioinformatics community, and simply saying, okay, here's the data, look at it. I think that we could have come up with a lot of interesting solutions there. I think that would be of benefit for everybody. And the sharing of data is, of course, complicated, and I'm fully aware of the Nagoya Protocol, and of course say that what if the sharing of data, not exporting data, also goes for sequence data, and not only, physical samples, but that's something that I think Article 4.2 actually fully takes care of. But of course, national governments could, of course, take restrictions there.
Okay, thank you, Frank. And then Mark, you want to make?
So I'll be very brief because I think everybody knows how I feel about this. But I think the main reason we're here today is to look at where public-private partnerships can be the biggest benefit. And I really see them in 3 areas. Number 1 is biobanking. So preparing biobank consortia where there can be instant or quick access to whatever biologic specimens are necessary to validate diagnostic tests. Whether that means including other viruses or other pathogens in order to make it a specific test or whatever. But we have big problems. You heard about Brazil. It's the same problem in China. You can't export serum or any specimens from China. You can't export from Brazil. There are many, I can go country by country, there are many regulatory problems in terms of doing that, but we need biobank access. Number two is we need lab capacity building and lab support, because even if I develop a test tomorrow, we need the labs and the personnel and the education to run them. So there has to be, we can prepare in advance. We can create an African network of labs, which we're trying to do now. We can create an Asia-Pacific network of labs through our partners, through Pasteur, through FIND, et cetera, in order to prepare the labs to do the diagnostic tests when we develop them. And number three is regulatory. Regulatory right now is a mess. So countries say, no, I need FDA, EUA approval before I get it. No, I want CE marking, even though you've got WHO pre-qualification. No, I need pre-qualification from WHO, even though you've got EUA. It's all over the place. Every country is different. It is a major headache to get the tests into those countries to actually to work on the patients. The patients are waiting for them, and the governments are blocking it. So I think there has to be some kind of preparation in advance, and that can be done with a PPP as well, including government and NGOs together. So I think those are the three. I'm a very concrete, action-oriented person. I think those are the three where PPPs can really serve a purpose.
Great. Clear points made by the panel. So the need for more communication with public health, the need for reference labs in the different regions, identifying the clear role between the public and the private, importance of data sharing, and setting up the biobanking and sorting out the mess of regulatory, if I can paraphrase the last point. So what is your, anybody has questions or comments on these points and specific for diagnostics? Yes, please.
Ronald from Sanofi. So I have a question for Mark. I hope not to be instructive. Mark, you know we believe in private-public partnerships, especially for this biopreparedness. However,
Can we have the micro, please?
The public sector or the funder are asking, so what's your estimated cost to develop the vaccines according to phase? Do you have some ballpark figures on what it takes to develop a diagnosis for such as these fancy diseases? And so that I think IMI2, CEPI could add that on top of our vaccine development costs, which are more or less known.
So it's really variable, but as a general rule, to develop a Me Too test on an existing platform for diagnostics is anywhere between 2 and 5 million euros. All right? To develop a novel test, okay, on an existing platform, a novel combination, novel antigen, et cetera, never been done before, no primary materials, it's around 5 to 10 million euros. Okay, we're not talking about timing. To develop a totally novel diagnostic platform, which is not usually required in emerging infections, is anywhere from 50 to 100 million euros. All right, and the timing is nucleic acid tests can generally be developed in about, I'm not including the regulatory deployment, et cetera. I'm talking about validation, the processes, et cetera. If I can instantaneously mobilize people and budget, a nucleic acid test can generally be developed within about a year. Okay, a little bit less if we really work fast under emergency conditions. An immunoassay is longer. We're talking generally about 1.5 to 2 years, depending on how it extends, you know, IgG, IgM, et cetera, and then other tests depends. But I mean, those are general ballpark figures, but they're very variable. And again, it depends on if you're talking about Ebola, it's access to BSL-4 labs, and you can add all kinds of costs and delays and things like that.
Yes, Michael?
Thank you, Michael. Thanks for the very insightful presentations and the discussion that is going on. My question really relates to what you've alluded to regarding having reference laboratories, having biobanks. These are really important things to do. But I've also been in a unique position where I've been involved in strengthening ethics and regulatory capacity in countries on one hand, and strengthening the local capacity for laboratories in countries on the other hand. And what follows is that as countries have more exposure, as their capacity for ethics and regulatory is developed, they tend to be more strict, especially when it comes to material transfer agreements, as when it comes to moving out any clinical research materials out of country. But at the same time, we know that there are certain things that may not be able to be done in country, and the need for reference laboratories. So, coupled with this challenge, what can be done on the side of diagnostics to circumvent this challenge? Recently, there have been good developments on the side of the work that is going on, the minimal invasive autopsies, for example, where they were also having challenges in moving materials from one country to another. But they've managed to circumvent this by strengthening the imaging technologies, whereby they're able to capture the problem in country and share it with expertise out of country without moving out the materials. Is there anything that can be done in the field of highly pathogenic, infectious pathogens to address this problem? Thanks.
Yes?
Well, there are, in terms of ownership, there are two types of samples, if I may say. These are the human samples, and the other one are any sample of any biological material outside of you, which is regulated by the Nagoya Protocol now. It means that this states clearly that the material which is coming in one country, which is growing in one country, belongs to this country. So if there is a plant which is growing here, it belongs to me. But this has extended also to pathogens. So you wonder why people want to hang on their pathogens, which anyway go away without you can say anything. But this has generated a lot of discussion. Also, at the time, some of you may remember the question of sharing of samples for influenza. This was a long discussion about sharing of benefits in exchange of sharing of samples. So we need to take this into consideration. So what we are doing now to try to address this, for at least for biological samples, not human samples, is we are working on a material transfer agreement development tool that would be could be used by countries when they want to see what it is that they want to claim. So it is they need to be some some comfort for the authorities of one country that when they authorize export of a material, they have looked at all the options and how they are exerting their ownership of the samples without overstating the value of a sample that they want to export. So we think that this might help. We should have this available at the beginning of next year, might help for the non-human samples. Now, for the human samples, of course, the serum and all that, this is an whole other question. And this is why we think that it might be better to have the tests done in countries rather than trying to export them.
Thank you for that comment. Any other questions? Please.
Hi, Nigel Silman from Public Health England. Just really like to comment on the biobanking side. I was quite heartened when biobanking was mentioned as one of the necessities going forward. Just to put things in perspective, Public Health England is currently holding a whole bunch of human samples from the Ebola outbreak from Sierra Leone, Guinea and Liberia. Now, when we actually approached the funding bodies for support to maintain this biobank for the greater good, it was turned down flatly because the funding bodies refused to pay the costs of working with these samples at BSL-4. So Public Health England is a UK government body. We're enjoying huge budget cuts like every other government body around the world, and we were left with the bill for actually doing this, and it cost us, just to put things in perspective, £100,000 for every shipment of samples that came out of West Africa. So I just wondered if the panel would like to kind of take a view on whether centralised biobanks, are really something that we should be doing. I believe they are, but I welcome your views as well on this.
And maybe also the potential role of the private sector in that specifically. Anybody wants to comment? Yeah?
No, but I think that at least to some extent that should be taken care of the mirror infrastructure and the EVA European Virus Archive project. So but of course if they do not have the funding to include your samples, then yeah. But I think that I would completely agree that that's really important. Of course, you could always debate for how long time we're going to continue to have are you going to have a biobank of several 100,000 Ebola samples, or is it sufficient to have a more limited number in the long run? I think that's a question. But a central biobank, or a few central biobanks for the world, seems to be very appropriate.
In my way of thinking, a biobank is like a website. Anybody can create it. You can create a biobank, you can throw whatever you want into the freezer. The real cost and the real problem with a biobank is, well, annotating it, it's maintaining it, it's the cost of running it, okay, having it available when you need it, et cetera, et cetera. And that has to be an ongoing cost. There has to be a curator, there has to be participation, there has to be a pre-agreement that who's got access to it, who's got rights to it, et cetera, et cetera. And that all has to be worked out in a consortium, in a partnership. Okay, whether you belong to EVA or not, et cetera, these are all issues. It's great to belong to EVA, but if you haven't got the money, either from them or anybody else, to support the biobank, then it's really not good to anybody, not to us, not to an academic, not to anybody else. I mean, I think you brought up EVA. It's a good example of EVA came to us a few years ago and said, we can supply, we can develop a test. As you heard, you can sequence a pathogen now in 48 hours. Okay, that's fantastic. You know what the pathogen is. You can create a nucleic acid test. You can primers and probes in about a week. And so EVA was supplying reference labs and everything, and they said, but now we've run out. We have no money. We can't supply. We've got requests one a day by email for tests. Okay, we can't supply them. We don't have money. We can't industrialize. We don't have GMP. We don't have distributors in these countries. Can you help us? So I think there's a lot of partnerships that can be done where everybody's got a strength, everybody's complementary to everybody else. bioMérieux cannot go into an area and sequence a pathogen in 48 hours. Okay, an academic lab can do that. I mean, the CDC can go do that. EVA can go do that, partners. We can't do that, but we can industrialize where they can't. So I think that everybody's got a role, and the biobanking is key to be able to do this. We need an agreement as to what specimens can be validated and can be used. We don't have an agreement right now. We don't even know if we need clinical specimens. So here's an example about how biopreparedness means we can all get together and agree on certain norms and standards. Right now, the FDA will accept an EUA submission without any clinical specimens if your level of detection work is good and your QC is good and your validation studies are good. WHO pre-qualification, emergency is a little bit different, but they require validation and on clinical specimens, et cetera, with some exceptions. Everybody's got rules all over the place. So I think if everybody agreed on these types of things, then there's an element of trust, and it gets to the research and ethics. If everybody trusted that the process was good, then the governments would trust that the study can go on without going through months and weeks of ethics approval, et cetera. But there's that element of trust that's missing. We don't have standards. So.
In terms of biobanking, I think this is a critical issue indeed. And Ebola, as you say, there are thousands, tens of thousands of samples all over the place. Public Health England is one. There's also, in Germany, in the US, and there are a lot remaining in the three most affected countries. So we've tried to see what we can do to help them, because they want to hold to their sample. Because again, this is ownership. So what can we do in terms of helping them preserve them? We have a study that we have conducted to identify all the samples where they are. We'll publish it soon. But what we see in the affected country is that there is usually these things, most of them are negative samples. So if somebody would have a time to go through them and look at what it is, they should be thrown away. Okay. And a few samples are of value, but already the work just to sort them out is already something that costs money. And then they are kept usually in one freezer. There's no backup. There's nothing. As you know, also the biosafety is not ensured. So what do we do in terms of global community to help this country build something which is sustainable? It could be a virtual biobank where it's stored in several places, but all this needs money. And as you say, there's no money. Nobody wants to support that. So it could be a public-private partnership, and it would be good to have a private entity which is provided resources to manage the biobank. But for the time being, public or private, there is no fund.
Yes, please.
So one question, I guess one comment. There's definitely, I think the two introductions were great, and in a way they illustrate the difference between kind of capacity to detect and surveil, and then actually the clinical diagnostics in a messy situation. But I know now that there is an initiative from the US, including USAID, related to some sort of a metagenomics approach to trying to capture potential risks, viral risks in Africa. Is there a European kind of actually trying to a full genome sequencing approach to trying to identify potential zoonotic viral diseases? Is my understanding. Is there a European involvement in that, and to what extent can we use more or less the similar method? So that's one question. My other question is related to where do we draw the lines between the responsibilities of the research funding system and the public health system in these issues? Because a lot of what has been mentioned now I see as a kind of responsibility first and foremost for public health authorities to actually fund. So where should public health authorities step in, and what are the roles of research funders compared to public health authorities? That's quite a blurred landscape, would like to hear your comments on that.
Yes.
Yes, I'm not sure exactly which major project you're talking about, but there are a number of pathogen discovery projects also funded by Gates. And I think that I actually personally have a bit of a problem with most of these projects because they're very much built around the way that you go out. You have a number of people coming from North America or Western Europe traveling down to Africa. They'll take minimum invasive samples from these 5-year-old kids that have died, take the samples home to North America or Europe and do the metagenomics. And I'm not really seeing that as a way of doing local capacity building in the African countries. So I think that I would much rather see it was actually if you set up that kind of capacity locally and then share the data. First of all, it will give the local capacity building so they could do something in the long run. But it will also enable you, at a later stage to much earlier detect something new based on clinical diagnostics. This is, I had a long discussion around this with there was an interview regarding this with Gates, and the same comments that this is very much focused on, well, we're going to make 5 Nature publications 3 years from now, writing history. So I'm perhaps a little bit negative on this aspect. I don't think that is helping the sub-Saharan African countries. And then the public health, what's the role? I think that the funders should perhaps consider funding into creating platforms for generating data and sharing data. I think that would be important. Whereas the public health, well, they are more what you could call data consumers. They should look at the data and also critically look at the data and give advice based on those data to what should be the public health measures locally, and of course in the case of ECDC and the World Health Organization more globally.
Yeah. Because I have
on this project, and
then we'll come.
Just on this USAID project, because this is something which has also come to my attention recently. What they would like to do is sequence if this is a virome project, they would like to sequence 99.9% of all possible viruses on Earth. Okay, and the idea is based on the Genome Project. But what is not clear to me is actually how much this will actually help in preparedness, because we had the sequence of Zika, we had the sequence of Ebola, and nevertheless you can't develop vaccines and commercial diagnostics against all possible viruses in the world. So, but the expert, I don't know what you think about it.
Piotr, and then we'll go to the questions.
You asked about Europe, and I'm not talking about metagenomics here, but even for introduction of the whole genome sequencing, there is great diversity in the capacity of the countries. And of course, we are aware of this coming, and we try to give some guidance to the member states, those especially who have lower capacity. So recently we ran an expert group, I think it was called NEXT, from the Next Generation Sequencing, and I can send you the report. They try to set some pathway for how to tackle this. But we have countries which sequence some pathogens already on almost all samples, and we have countries with no capacity for sequencing, no capacity for storing the data, no plans for how to analyze the data. So we are trying to come up with some guidance in this diverse situation in Europe at the moment.
Okay, so we have 2 questions. We'll take both of them.
Yeah, thank you, Herman Gust, University of Antwerp. I want to come back to the issue of biobanking. And this is a comment out of sheer frustration, because I think we've missed so many great opportunities in Europe, for instance, to let's say, develop and maintain biobanks, because we've received a lot of funding from the European Commission for a lot of projects, like GRACE and MOSA, Saturn, and Diagnostics, and I can give you many more, where we collected thousands and thousands of specimens from patients linked to fantastic clinical information. And then, of course, the funding comes to an end. The mechanism of funding for the European Commission does not allow us to maintain the biobank. And then these samples are sitting in the freezers. Sometimes they're lost. Sometimes there's nothing that happens to these samples. And I think it is actually unacceptable to collect all these specimens from patients without having a vision of what we're going to do with them. And I think it's really, really, really frustrating to see this. So my point is, and it's an invitation to the diagnostic industry, what are you waiting for a proposal to maintain these biobanks, Mark? If you want your samples for your validation of the test, you're very welcome to have them. But why don't you make a proposal of how we can maintain the biobanks and how you can co-fund them so that we will collect them through the public funding and then you will have access to them to the private funding?
Okay, we'll just take the second question first and then we'll go back to the panel to finalize the discussion.
Jean-Christophe Audonné, Merial, Animal Health Division of Sanofi. I have a question for Mark Miller and maybe also the panel. How do you accommodate today with the new requirements from Nagoya Protocol to access to the pathogens and genetic resources? So if you do just diagnostic, it's not under the protocol, but if you do development and developing diagnostic kits, commercial kits, you are under that and you have to discuss with countries of origin. So is there a system that you have defined or designed to access to these resources? It's true, and impacting both diagnostics and also vaccines.
Okay, so two questions, one on the biobanking and one on the
So, in response to the biobanking, have the diagnostics industry done what they can in order to establish biobanks to be able to facilitate things? No, we haven't. On the other hand, it's an imperative, sine qua non before moving on to validate any diagnostic tests that are developed. So, I think that it's a very good point, and I think these are the kinds of things that within IMI, we've got a project on the table that we'd like to propose called DRIVE-DX, where we're looking at the big obstacles for developing diagnostics. It includes for regular pathogens and emerging pathogens, and biobanks is one of those issues. And if biobanks is one of those issues and it needs addressing, then I think it needs a common a consortia-like agreement where everybody knows what we need to do for biobanking and where we get the specimens and who's going to pay for them in perpetuity and where they're going to be frozen and who's going to curate them and all the things we talked about. Freezing the specimens are easy. All the rest of it is the hard part. And for the people who are not diagnostics makers here, even the number of times that you've thawed the tubes is important to us. So if you thaw them too many times, okay, we can't use that specimen anymore. So it's a simple practical things about how many aliquots of a sample and where are they kept, like you said. Is there a backup? How many times has it been thawed and refrozen? Those are really important things for validation of a diagnostic assay. So this is where all the cost is. And yes, we haven't done our job. But then again, I think the recognition of the importance of biobanking is something that's really only recently come to the front. So that's that. I didn't quite understand the question about access to the pathogen, so I can't really answer. Maybe we can take it offline. I don't really, unless you want to explain.
Yeah, you want to reiterate?
To get the approval of the country of origin when you sample a resource, a genetic resource, including pathogens, including microorganisms, and there is no exception except pandemic flu in people. That's the only one.
But how to circumvent?
And there is no mechanism today which is in place to access to that,
yes.
to get the permit.
Frank, you want to?
I don't think that anyone has any easy solution in terms of the interpretation of the Nagoya Protocol. But if you look at what has been written, and I know that the different translations are not exactly the same, but then actually, if you look at Article 4, if you collect information and you share it openly without having any personal, financial profit making out of it, it's for global public health, then you can do it without contacting any of the national contact points. So it as far as I can see, and that's also I know the chief advisor from foreign protocol from Harvard, I think, or Yale, actually agrees that this should be the interpretation. But if you do that as an industry, a single company, and you make profit out of it, well, you cannot take the information, anything out of a country. But you can actually take samples, and as I said, we have just done it, and you do not need to contact anyone regarding the Segoe protocol locally. Even Brazil, we have samples from Brazil, but as long as it's for the purpose of sharing this openly without no personal profit. But of course, it's going to be interesting to see if you get sued by someone.
Okay, thank you. I think we will have to close this. I just want to see if there is any final remark from the panelists. Marc, do you have any final?
I just wanted to address the comment that was made about whose responsibility this is, whether it's public health or researchers or whatever. And I would love an answer to that, because the next question is, as an industrial, who do we work with? So do I work with public health? Do I work with the government? Do I work with the academics? Who do I work with so that we can move things along? Because if I work with the WHO, then Guinea says to me, oh, you didn't work with us. You didn't work with the government and our public health agency. You worked with WHO. They don't speak for us. If I work with Guinea, then WHO says, well, that's not us. Okay? If I work with somebody else in the United States, public health, whatever, CDC says, oh, you didn't work with us. We can't bless you. So if you figure out who is responsible for this, then I would love to know, because then I'd know who to work with.
Okay, clear. Frank, any final comments?
No, I think that's, well, this is new for me to be here, and I think that hearing this, I think that at least with our project, we are still on the right track, but I can certainly also see that we probably need to embrace a little bit more, a whole new community. And I think that will be hopefully interesting in the years to come.
That would be a very nice result of this panel session, actually. Piotr, any final comments?
To me, the key lesson is, ECDC operates a number of disease networks under for the EU level surveillance under the Decision 1092 on serious cross-border threats, and many of them, or most of them, have laboratory components, so there are laboratory sub-networks, and there the people get together to discuss the current issues and upcoming developments, and I think there is a need for a better link with IMI initiatives, actually. So I think I'll take it with Angela later, how to ensure this link both ways, so that the networks hear about all the developments that are going on, and then maybe they can advise also for the future plans.
Great, that would be nice.
Well, I think that what's important is that the value of diagnostics is more and more recognized in terms of a tool for public health, for emergency diseases. But this being said, the diagnostic industry and the regulation of diagnostics is still it's not a mature field. So there is a lot of things to be done in terms of harmonizing between countries, in terms of seeing what are the appropriate pathway, and I think this is something that should be taken up with urgency, as was said by many.
Thank you. So clearly wonderful to have a diagnostic session in this day of meeting. There are clearly opportunities for public-private partnerships, whether it's on biobanking, whether it's on the regulatory issues, and I think I don't want to look too much in that direction, but it's true that having that ready in advance can save a lot of time at the time when things have to go fast, and so I think that's a really key point. And then obviously a good dialogue between the different partners, the different types of stakeholders is essential. So, and then within that all, need to clearly define also the roles of every type of partner. So, a great panel. Thank you very much to the panelists and to the audience, and bon appétit.